All right. Welcome to our session with Sarah Teichmann, our keynote speaker for today. Very proud to have Sarah with us. Everyone in the single cell world knows her very well from her research and from her leadership of the Human Cell Atlas Consortium, which she and Aviv Regev kickstarted a while back. Sarah is so she's head of cellular genetics at the Wellcome Sanger Institute, did her PhD at MRC, Cambridge, UK, and was a Bate Memorial Fellow at University College London. Then she started her own research group at MRC in 2001. In 2013, she moved to the Sanger campus in Kingston, Cambridge, and she was joined between EMBL EBI and Sanger. Sarah is an EMBO member, fellow of the Academy of Medical Sciences, and she's received a number of prizes, including the Lister Prize, Biochemical Society Colworth Medal, Royal Society Crick Lecture, EMBO Gold Medal, and her work is at the intersection of immunology and genetics, and I think even before single cell, worked in protein structure and became famous there. And now is really one of the preeminent single cell researchers in the world. A real privilege to have you as our keynote speaker, Sarah. Thank you for accepting.
Thanks so much, Shyam, for that kind introduction, and thank you for having me here. I'm going to share my presentation now and just make sure that this works. It's incredibly exciting to be presenting here at this HCA Asia meeting. And, I'm so pleased that there's a large community that's been able to meet online and come together in Asia, so to speak, in this virtual space. And I just want to say a huge thank you to Shyam, Alistair, Miho, and Jay, and all the others who've contributed to putting this program together and to making all the logistics work together with the broader program committee. And it's a great pleasure to talk about human development and two pieces of work that are largely unpublished at the moment that I want to share with you today. And the thinking, basically, the common sort of conceptual thinking behind both of these projects is that we're now at the point where we can really assemble or move forwards in getting to a point where we're getting a broader assembly of human development in terms of cell lineages, in terms of where cells, where and when cells are migrating during development, to put together in a more comprehensive way our characterization and our understanding of how a tissue forms. And the methods that underpin this are, of course, genomic methods, but also data integration of existing data sets without further data generation, looking at the same time point, but different omics modalities or different tissues, and the same omics modalities across different time points so that we can really understand development over space and over time. And here I want to introduce this using two different projects that focus on two different developmental processes. The first is development of immunity, and Shyam mentioned that I have a long-standing interest in this, in the immune system, both innate and adaptive. And of course, this is a distributed system encompassing both lymphoid and non-lymphoid tissues. And so the approach that we're taking here is to integrate a lot of existing data together with new data from different tissues and different time points to learn new things about development of the immune system. And so I'll focus a lot on methods in this part, in the first part. And then the second part, I want to talk about the development of a structure, one continuous structure, and that is the limb, and integrate data from single cell and spatial across different time points to see whether we can trace the formation of the limb and the digits in human development. So for the first part, we're asking when, where, and how do immune cells develop? And for the when, basically, we have here a kind of overview figure from a review from Mas Hanifa and my groups together last year, where we've got first trimester, second trimester, and then birth and postnatal development. And you can see that in the early first trimester, we already have innate immune cells developing, macrophages, mast cells, and then monocytes and dendritic cells, and then later, already sort of in the second trimester in the liver, then the neutrophils, eosinophils, and basophils come, sorry, in the bone marrow, not yet in the liver. So this is later in the second trimester. And you also start to already get immune cells migrating into the periphery, so into non-lymphoid tissues in first trimester. And then in terms of the lymphoid compartment, you've got the innate lymphoid cells, NK cells, and unconventional T cells and early B cell progenitors developing in the first trimester. And then as the thymus forms, you've got a more mature T cells developing later in the first trimester, and ILCs migrating into the periphery, intestine, liver, lung. And then it's not until birth that you get really T cell dependent B cell response with germinal centers, the full repertoire, and so on and so forth. So it's really, this system is really interesting and complex because the development takes place over the whole, essentially the whole of pregnancy and even postnatal stages, and includes many different tissues. And those tissues are shown here schematically, including the yolk sac and the AGM, aortic gonadotropin nephros, where you have those early macrophages developing and then migrating already into other tissues. Then the liver basically becomes our major site of hematopoiesis in second trimester, in first trimester and early second. And so the liver is kind of our bone marrow before the bone marrow exists and becomes the major site of hematopoiesis. And it's after that, sort of around 7, 8, 9 post-conception weeks, that the thymus develops as an organ. And of course, the thymus is where we get T cells developing. And cells migrate from or exit from the thymus and bone marrow and liver and go to the spleen, as well as to the peripheral tissue, the peripheral organs, the skin, the kidney, the gut, the lung, and so on and so forth. And so this is a really complex system that we want to understand in terms of when the cells originate and then how they migrate and contribute to the development of other tissues. And together with my partner in crime on this project, we've had a collaboration over 5 years now with Mas Hanifa's group. So it's been a tight partnership where we've studied the fetal liver, the thymus, the bone marrow, and innate or a peripheral immunity, kidney, gut, and so on and so forth, and focused on the intrinsic changes of hematopoietic stem cells in these different compartments and over different time points. So how do the progenitors, how does the personality or characteristics of the progenitors change over time and space, and how are they influenced by their different niches in the different tissues in yolk sac, AGM, liver, bone marrow, and so forth? And how does that change the potency of the stem cell when it's in these different niches? And how do cells adapt to organs in the periphery and contribute to their development? But we're at a point now where we're at a kind of interesting and exciting juncture where we have all this published data where we've focused on individual organs to a large extent, although already asking questions about how stem cells and migrate and change and so on. But we can now sort of fill in gaps where we don't have comprehensive data such as the spleen, and here we've generated new data on the spleen. And this is with Mas Hanifa's group and ISAC Go specifically, We can then dissociate the tissue and study the cells in suspension in terms of gene expression, but also the different VDJ repertoires, alpha, beta, gamma, delta, and B cell VDJ repertoires of the antigen receptors, and and then integrate close to 1 million cells across 220 libraries and 25 donors, and include also Visium data that's shown here for 2 time points in orange, in the second trimester, about 15 and about 18 post-conception weeks, where there are stars. This is spatial transcriptomic data at 50 micron resolution. And you can see here the donors at the different time points, the different gene expression libraries in red, and the VDJ libraries in blue, green, and purple. And so this gives us, in aggregate, we're starting to get a relatively comprehensive picture across different tissues and different time points. And I want to spend a little bit of time talking about the analysis pipeline, and this is put together in this schematic by Emma Dan, who's an incredibly talented PhD student across my group, and John Marioni, and just spend a few minutes focusing on methods instead of sort of just skipping over them quickly. So I've already talked about integrating the data with SCVI and sort of deep learning framework, and then with Milo, which is a graph neighborhood focused approach, we can calculate differential abundance of expression patterns in cell neighborhoods between conditions, and in this case it's between early and late development in different organs. So we can ask, in the liver, is there a difference in the neighborhood because of changes in cell state, for instance, in progenitors in early versus late development, in the erythroid lineage in early versus late development, and so on. And Milo is a differential abundance testing framework that we published in Nature Biotech earlier this year, and I encourage you to look at the GitHub and see if you find it useful. So it's an unbiased search basically for changes across different cell states. We can also use factor analysis to extract tissue-specific expression modules, and so this is basically where we're showing, let's say, pro-B cells, so a particular state at different developmental stages, and show that they're expression modules that are specific to basically a stage or an organ that's not found in others. And so basically, class 2 expression is higher here than here, and so on. And I'll spend a few minutes also focusing on features of the antigen receptor repertoires in T cells in this talk. And what we're using here is a combination of the 10x Genomics scripts, Dandelion, developed by Kelvin Twong with Mena Clatworthy and me, and then also bespoke scripts for this purpose. And what I mean by that is basically the combination of individual segments from V, D, and J that are being used in a cell population, basically assembled together. Another approach that we're using is query to reference mapping, and in this case we're using SC Arches from Fabian Theis group from Mola Fohahi. It was published earlier this year, where we're integrating, for instance, that big fetal data set of 1 million cells that I mentioned that we've assembled here, as compared to immune cells in adult. And so we can see which regions of the fetal which regions of the fetal UMAP are occupied, as it were, by the adult cells. And finally, I mentioned earlier that we have Visium 10x spatial transcriptomics, and so this is where you've got the 50 micron voxels of transcriptomes across a 2-dimensional tissue section as a grid. And what we can do is then use cell to location as a deconvolution framework for identifying basically which cells are predicted to occur in which proportions, in which voxels of this landscape of gene expression in the tissue section. So you can see the liver here, where we've got hepatocytes fairly evenly distributed. We've got erythroid cells, kind of in more punctate pattern in the liver, and then if we move to the spleen, we can see little lymphoid aggregates in patches around. Macrophages more evenly distributed, and for the thymus, we can see the medulla, which is where the medullary thymic epithelial cells are sitting in the center of each lobe, very clearly with these bright patterns here, and then the cortical pattern, which is the edge of surrounding each lobe, which has a different type of epithelial cell here, and then what's suddenly very clear, which is something that you miss from suspension cell data, is that this sample has an extrathymic lymphoid tissue, so a sort of mini lymph node basically stuck at the bottom of the tissue section here, and we can pinpoint and localize that very clearly and see it. And so that's, the power of this spatial data is really, very, very clear, even though it's not at single cell resolution, and we're getting to a sort of pseudo single cell resolution here. And so for this project, I just want to focus on one sort of biological highlight, where we're asking when, where, and how does unconventional T cell development occur? And unconventional T cells are all belong to a variety of different lineages. They're the alpha beta T entry cells that exit from the cortex, which is that tissue that's surrounding each lobe in the thymus, and then entering the medulla. So they are the ones that have undergone the positive selection. They have correct alpha beta T cell receptor protein chains that form a complex, and they are then entering the medulla for negative selection. And for sorting out which cells recognize self versus non-self, and getting rid of the ones that recognize self. And these cells, as well as Treg, CD8 alpha alpha cells, innate-like T cells, Th17-like cells that have more CCR6 and so on, NKT cells, and so on. They're all characterized by ZBTB16, which is a transcription factor that you can see highlighted here in these populations in the UMAP of the T cell compartment over here on the left-hand side of the slide. And so here you can see the subpopulations that basically have that expression. And if we take these together, all of these cells, and look at the aggregated data across all the different organs, we can see a common pattern that the percentage of these cells is much higher in early development in all of the organs, the percentage of these cells out of all the T cells, as compared to later stages of development where they sort of decrease in proportion as the conventional T helper and T killer cells increase in proportion, both in the thymus where they're made, the cells are made in the first place, as well as in the other organs where the cells are migrating to. And in the thymus, you can see this very clearly, where when the thymus is formed, like as I mentioned, 7 to 9 post-conception weeks, the epithelial and stromal cells that make the organ are kind of coming together. And in those early stages, as it's being formed, we've got a dominance of these unconventional T cells, whereas later they form a minor fraction. It's the conventional killer and helper cells that dominate. And if we then drill into, when we aggregate this data, we have enough data for the first time that we can, for these more rare kind of smaller populations, we can aggregate the data and then understand for the first time how the features of their T cell receptor repertoires differ from the conventional T cells. So this is something that's only possible now. This wasn't possible, for instance, when we first did a cell atlas of the human thymus, which we published last year. What we see here very clearly in the unconventional T cells is that they have a preference for usage of the segments that are marked in the pink stars here. And you can see that these are clustered around this area, which is the junction of the V genes versus the J genes. And so the junction is here. And these are basically segments that are close to each other in the locus, in the DNA sequence, in the one-dimensional DNA sequence. And so these are the most likely to get recombined first, the proximal V and J genes, simply because they are sort of closest to each other. So just kind of by the stochastics of the enzyme randomly bringing together different segments of the DNA, this is most likely recombination of the RAG enzymes. And then as over time, as you get subsequent rounds of recombination, the distal segments that are here at the 5' end of the V and the 3' end of the J are more likely to get used. And we see this being more enriched in the conventional CD4, also Treg and CD8 lineages. So killer, helper, and Treg lineages have a more even usage of the different segments that includes the segments that are further away. And you can see this plotted on in terms of PCA space, where we've got the progenitors, the double positive, and so on here. But then over time, you've got migration to the killer lineage and the CD4 helper and Treg lineage. And the repertoires of those two are distinct from each other, but they are both using, having a more broader usage of different segments, whereas the unconventional NKT, CD8 alpha alpha, and Th17-like have a more restricted usage that's dominated more by proximal segments. And so that sort of suggests that there's different mechanisms of development for the unconventional versus conventional T cells. And one of the models for this has been, in the past, has been that you get unconventional T cells developing by TT cross-presentation, and conventional T cells developing by thymic epithelial cells presenting to the T cells. And this sort of really makes sense in terms of the dominance of the unconventional T cells early in the thymus, in the sense that at those early time points, 7, 8, 9 post-conception weeks, is when there are simply fewer epithelial cells. The organ is smaller because it's only just starting to develop into the structure with the lobes and the cortex and the medulla and so on and so forth. And so as the T cells develop, the likelihood of a T cell meeting another T cell, simply in terms of the sort of spatial relationships, is higher than later in development, where there are more thymic epithelial cells that have differentiated and matured, and they've there's simply a larger organ, more of them, so that the likelihood of a thymic epithelial cell presenting to a developing T cell is increased. And so we think that this TT cross-presentation is a mechanism, that's occurring because you have these proliferating cells that are bumping into each other, and if we look at basically an artificial thymus, so an organoid system in the dish, we think that there's a similar mechanism happening, and the reason for that is that this is a system where you've got hematopoietic induction, and the hematopoietic progenitor cells that can either develop, you can make them from iPS cells, or you can take a hematopoietic stem cell, which is kind of like what happens in the thymus, in the sense that the hematopoietic stem cell comes from liver or bone marrow, enters the thymus, and then it meets thymic epithelial cells that present it with Notch ligand, with a delta, and give it a kick, a signaling kick that sends it then down the direction of T cell development via the early stages. In this particular artificial thymic organoid system that we're working on, it's from Gay Crooks's lab. It's a mouse stromal cell, MS5, and the ligand is delta ligand 4, and that's a human protein that's engineered into the mouse cells. But they are not thymic epithelial cells. They're a kind of artificial stromal cell that's kind of a Frankensteinian mixture of mouse and human, and that's presenting this artificial ligand, for the mouse cell, presenting the human ligand to the human progenitor cell. So there's no epithelial cells there. There's these stromal cells there. And when we characterize the culture over time, 3 weeks, 5 weeks, 7 weeks, it's a long culture protocol, then what we see is that the space that's occupied in this UMAP sort of migrates from left to right. And if we want to understand the identity of those cells, then what we did was use those fetal data and those fetal cell states, and model them using logistic regression, and then do the scoring against the cell culture in vitro derived cells. And what we can see is that the left-hand side consists of the natural progenitors, double positive, double negatives, and so on and so forth, and then alpha beta T entry here as you're crossing over from the left side to the right side over time. And on the right-hand side, these cell states are really dominated by unconventional T cells. So we've got NK, NKT, CD8, alpha, alpha, and so on. So it's all these unconventional T cells and fewer of the CD8, CD4, Treg, and so on, which you'll see in a mature natural thymus. And this this makes sense to us because in the culture we know that over the time, say by 7 weeks in cell culture, the stromal cells are dying off, and it's really T cells that are left as viable cells in the culture inside the center of the culture. And there are no epithelial cells, obviously, because it's an artificial system. And so the way these cells are very likely developing is through TT cross-presentation. And so the in vitro system basically kind of confirms the mechanism that's likely to be leading to these unconventional T cells in vivo. And we've got a nice here sort of information coming from the in vitro system and telling us about the mechanism in vivo. And of course, the in vivo system will also give us clues as to how to improve the in vitro system to make the cells more faithful to the reality. And I want to stop here talking about development of the immune system and just focus on this last point where we've seen that we can get an integrated picture and a new understanding of T cell development by stitching together the data from the different organs and different time points to understand the features of these cells in terms of their cell states, their antigen receptors, and their developmental mechanism. And I just want to again acknowledge the people who did this work. I've mentioned Emma. Chen Chu is an amazing clinician scientist, PhD student in my group, originally from China, from Asia, via Singapore, came to Cambridge. Many other members of my group, also Menek Clatworthy's group on the Dandelion software, Mas Hanifa's group, who's been a partner in all of this data generation analysis, and specifically Isaac Go from her group, as well as other group members, the funders and the donors and their families. And if we're not out of time, I think we've still got time left, I'm going to move on from the development of immunity in this distributed system and how we've tackled this to a second story on limb development. And of course, this is a completely different situation here because we have one continuous solid tissue and structure that we're looking at and trying to understand how it changes in space and time. And of course, in the limb bud, basically, although it's a single structure and it may seem simple in that sense, it consists of a lot of different lineages and systems, musculoskeletal, vascular, neurodermal, and so on. And so you've got a lot of heterogeneity even within this single continuous physical structure. And basically we've got development from a simple initial bud, to this incredible complexity of digits, skeleton, musculature, and so on and so forth. And basically the understanding of development is important for a number of different reasons. Rare disease is the obvious one, but we've also got other musculoskeletal diseases, and understanding how to generate these cells in the dish using in vitro systems, like I just showed for T cells, is also relevant for many musculoskeletal lineages. So studying human development is really important and informative from from many different points of view, and I'll emphasize that again at the end. So in this case, again, we used a combination of different modalities: suspension cells, spatial transcriptomics, and both from the upper and lower limb, and from studying it from proximal to distal, which is also the direction of development in this structure is from proximal to distal. So you've kind of got a temporal progression in space at any one snapshot in time, and then disaggregating the cells and studying the total cells, as well as tissue sections from the time points in first trimester. So it's from 5 to 9 post-conception weeks. So it's all within the first trimester, where all of this development is happening very quickly. And the questions that we're asking is, what is the cellular heterogeneity and how does it evolve in space and time? What are the key molecular mechanisms driving cell type specification and limb morphogenesis? And what's the conservation between us and rodents? And what we worked on here is gaining a high resolution atlas of the embryonic limb development, so that we can identify many different substates in the muscular lineage, but as well as cartilage, so osteochondral, fibroblasts, so sort of the stromal cells, tendons, tenocytes, and other lineages. And it's worth saying that these all come out of the lateral plate mesoderm, which is essentially the early stromal progenitor. And then this develops into this multitude of different subsets of lineages that I've just mentioned. So it's really quite an amazing diversification process that you can see here that's all coming out of these progenitors. And basically over time, you've got a decrease in that central progenitor population. You can see there's hardly any left here at 9 post-conception weeks, whereas it's very dominant at 5 post-conception weeks. And but you've got more and more of those mature states that are in the outside of the UMAP that are developing out. So basically the muscle here, the tendon, cartilage and bone, and so on and so forth. And if we integrate the single cell and spatial using cell to location, which is again the deconvolution framework that I mentioned, that's in press in Nature Biotech, and it's on GitHub, and it works really beautifully also for rare cell states in our hands. What it gives us is the spatial distribution of cell types. And so what you can see here is basically the different zones in digit development, chondrocytes, prehypertrophic chondrocytes, and so on, occupying very clear regions in this in the hand and foot plates. And in order, another point that I want to emphasize is that later in development, obviously the limb is growing and maturing and simply taking up a lot of space. And so the way we had to stitch this together was using multiple regions, each put separately on chips and then stitched together. And what this allows us to see or kind of reconstruct beautifully is the different regions of the cartilage and muscle developing. So here you can see perichondrium around the outside, and then osteoblasts kind of more inside, prehypertrophic chondrocytes at the junction. So these are chondrocytes that are becoming bone, and then the cartilage, the collagen from the cartilage, and so on here. Articular chondrocytes again here at the joint junction, and perimysium is also very clear around the edge. So there's even perineural fibroblasts that we can distinguish. So it's really it's quite exciting to be able to reconstruct these developing bone and cartilage features in this quantitative genomic way at the molecular and cellular level. And so it's giving us basically the evolution of these lineages. There are these 10 major lineages, but within them there's 55 different cell states from 5 to 10 post-conception weeks. And within them, there's many that were known before, but basically atlasing them in this way at full transcriptomic breadth reveals a lot of new factors and mechanisms driving cell type specification and even new development pathways and new cell states that I'm only going to be able to talk about some of them here. So if we focus on the mesenchymal compartment and the changes in abundance over time, we can see here the mesenchyme progenitor from the lateral plate mesoderm dominating early on, and then we've got obviously the outer zone around the digits early on that's lost later because there's the death of that sort of webbing between the digits that happens. There's you've even got dermal fibroblast lineage and perineural fibroblasts and so on, perimysium, perichondrocytes developing out of those progenitors, and then it's really the mature cells by 9 post-conception weeks that are present, and the progenitors are completely gone. And over these developmental trajectories, we can obviously define different transcription factors that are controlling, or that we predict to be controlling, and some of them we know from the literature, these different lineages in the different compartments over the different stages. And so here, these are different transcription factors here for the mesenchymal, PZC, chondrocyte in green, interzone and articular zone chondrocytes sort of at the joint, osteoblasts kind of for the bone, tenocytes, a tendon here on the right-hand side, and the key transcription factors that are coming up in these lineages, marked here on the bottom in the different colors. And we can also, in the 5 and 6 post-conception week spatial transcriptomic slides, we can show where these transcription factors are in the UMAP, but also where they're occurring in space. And so the progenitor, as I mentioned, there's development from proximal to distal, and you can see the progenitor, the early lateral plate mesoderm, for instance, and progenitor transcription factors kind of in the more proximal, the PZC kind of those around the edge of the limb bud early on, that then develops into the digits at the end, around the outside. The tendon isn't shown, but the osteoblasts and perichondrocytes are shown here very clearly, and so this is really a reconstruction. You can see here the chondrocytes also occupying where the digits will come kind of at 6 post-conception weeks, with those SOX9 transcription factor that's in the early chondrocytes. And these regulatory factors and the signaling factors that we can infer, that I'm not going into in detail, but from the niches in the spatial transcriptomic data and the signaling that's predicted to occur within each microenvironment, we can evaluate in vitro systems such as in vitro chondrocyte system set up by Sue Kimmer and Stephen Woods in Manchester, where we can assess to which extent the in vitro protocols are generating prehypertrophic chondrocytes versus osteoblasts. And if we deconvolute the in vitro bulk RNA sequencing data using our single cell atlas as a reference, then we can see that there's a significant difference between the extent of chondrocytes, for instance, in this differentiation versus this differentiation and this sample. And using and then make predictions about how to bias the in vitro cartilage protocol towards the chondrocytes versus the osteoblasts, for instance. And using our transcription factor activity by our in vivo cell types and data, we can, for instance, show that FOXO1 is basically high in mature osteoblasts, but not in the pre-hypertrophic chondrocytes. And what we want to do is bias towards chondrocytes. So therefore, we want to inhibit that transcription factor. And there's a small molecule that you can order online that does that. And so if we add that to the cell culture, then that increases collagen production and BMP activity and decreases the TGF-beta signaling. And so we've again, like in the in vitro artificial thymic organoid system, we've learned something from the in vivo data here that's useful for creating these cells in the dish and engineering which lineage they generate. If we consider basically now taking a step back, considering the entire sort of spatial transcriptomics landscape, we can define gradients during limb morphogenesis. And so it's, as I mentioned, the limb is known to develop from proximal to distal, but there's also anterior posterior axis, and we can define the molecules that are expressed in an axis along these axes, basically by mapping them over the two-dimensional grid. And you can see here in distal we've got Wnt signaling. In this more posterior region, BMP signaling and the specific transcription factors, patch1, HAN2, and so on associated, and TBX2 and 3, and sonic hedgehog associated with that. And so this really gives us a way of characterizing the spatial expression across these gradients, including HOX, where we can then define again quantitatively which how the HOX factors are expressed from proximal to distal, and also in this anterior to posterior segments, and map the gradients, but also using approaches like spatial DE or Spark and so on, map the zones basically that define the distinct kind of geographies of these gradients. I also want to spend a few minutes talking about digit morphogenesis, which is the specific kind of process happening in the hand and foot plates, where you've got the bones developing the center and then that interdigital space that I mentioned. And you can even see that from the histology going on very clearly, but you can also see it in terms of the transcriptomics, the distinction. And if we then map the differential expression between the interior and the exterior spaces, we can see that there are factors that promote digit survival that we can identify in an unbiased way, like IRX1, IRX2, and then the factors that are triggering the interdigital cell death that are in the outside but not the inside by calculating the difference. And then also link these expression patterns to genes that are associated with variations in limb phenotype in the human population. So there are syndromes like brachydactyly, and sort of non-syndromic brachydactyly, and syndromes that also have that, but also other features, and map the expression of these risk factors for those syndromes onto the spatial transcriptomics data. And you can see that the non-syndromic have very specific patterns, and the syndromic have more broad expression of these genes. Syndromes with syndactyly that have different features of the digits, you can see expressed here in the interdigital space, DLX5 and again, and so on and so forth. So we can learn basically by studying the molecules driving cell type specification. What are the program of transcription factors regulating cell development, and can we leverage that for engineering cells in the dish? What are the key players in axis determination and digit development, and can we link them to human syndromes and variations in phenotypes? Finally, I'm going to finish off by looking at the conservation between mouse and human, which is really important and useful, so that we understand where the mouse models are precisely mirroring our own developmental and organ and structural features, and where they're different, and also understanding, the evolution of expression programs, the evolution of regulation of gene expression in cell state. And so what we used here was multi-map, which is our manifold integration method, to take the mouse, published mouse data from us, but also, or unpublished data from us and published data from others, and integrate them with our human data. And this is the mouse data, I should say, is a collaboration with Barbara Wold's group at Caltech and Brian Williams. And so if we take the mouse and the human, basically they align them, we can focus in on also specific factors, because I should specify that with Barbara's lab, we also profiled the mouse going from proximal to distal in 3 sections. And we can plot basically which factors in the mouse and which ones in the human share that proximal to distal differential expression. And what you can just very simply see here is that there's a large sharing with where the red and the blue histograms are identical for many transcription factors. But then there's in particular non-coding RNAs where there's red only, human specific, for instance, or also blue only, that are more or organism specific and not shared. And so there's a general conservation of the key regulatory factors, but then there's variations specifically in things like long encoding RNAs, like this one and this one, and so on. And so to this last point, I'll just wrap up here by saying that we have access to determining gene expression patterns largely conserved, and then with small variations. And so for this second part, this is all unpublished work. I want to focus on the acknowledgements now. I've mentioned some people along the way, but really key members of my group are Peng He, who's a postdoc who did his PhD with Barbara Wold on the mouse limb development, and Sid Lawrence, who's a clinician scientist, an orthopedic clinician scientist in my group, who's been fabulous kind of in interpreting the data over the past year and a half or so. Many other members of my group who contributed. We collaborate with Omer's group and Sam Bishati in my department, also Liz Tuck from the support teams. And this has been a long-standing collaboration with a former postdoc in my group, Hong Bo-sheng, who kicked off all of this work. And he's now a professor at Sun Yat-sen University in Guangzhou, and it's Bao Sheng and Xiaoyu Wang from his group who are equally important and have done a huge amount of work. So this is an Asia-UK, a China-UK collaborative project. I've also mentioned Sue Kimber and Steve Woods from Manchester, Barbara and Brian from Caltech, Roger Barker and Xiaoling He, who are collaborate on the tissue acquisition in Cambridge, Michaela Storr from Cambridge, 2 geneticists, David Fitzpatrick and Helen Firth, and also Andrew Dean, and finally the donors. And I just want to summarize the whole talk, that's covered these very distinct, different types of processes going on in human development. Is in both sections, we saw that the in vivo data is sort of giving us a recipe for how to engineer cells in the dish. In both, we're getting insights into disease, basically by studying human development, including rare genetic variations. And we're able to make a detailed map of cross-species conservation by a deep study of both human and mouse. And I will stop there, say thank you, and take any questions.
Thank you very much, Sarah. Wonderful survey of 2 very different aspects of development. I encourage everyone to use the Q&A button to ask their questions. And while waiting for that, a couple of questions from me. Maybe I'll start with, okay, I'll start with the first part of your talk then. So the human cell atlas and immune system is a major focus of the HCA, the adult immune system, the developing immune system, and you've been a pioneer of that, and a few others as well. And you're getting richer with each new iteration. You're getting richer and richer maps of immune cell states because the technology has improved, the size of the data set has improved, and so on. But nevertheless, when you talk to immunologists, and of course you're an immunologist too, but maybe more traditional immunologists, they say, I want to see an antibody and I want to see a fax, and then I'll believe that that's a cell type until then, because that's how you define, immunologists define cell types. So is that paradigm shifting? Is the HCA succeeding in shifting that paradigm, or is that even the wrong question? Like, does the HCA believe it will shift that paradigm to more, classification based on a larger number of molecules than, the proteins that are usually tagged with antibodies?
That's a really philosophical kind of question, Shyam, and thank you for raising it. I think it's a great question, and I do feel that, I feel that it goes both ways, right? So I think we are shifting the community, and we are influencing the field of immunology. So the Cell Atlas community is actually kind of, influencing and changing what's accepted conventional, what's cutting edge and so on in those fields like immunology. And that's fantastic and a real credit to all of us. At the same time, having the protein level, which is really what the flow cytometry is, it's the protein level, that is informative. There's absolutely no question about it, because there are things like particular isoforms, post-translational variations, that whole level of control that's post-transcriptional and post-translational that you can only see at that level. And so the multi-omics modalities like the CiteSeq, I think, are really powerful in bringing those worlds together. So because those panels are, in the hundreds now, and so you automatically have that integration then across the different modalities, and that's, in a way, that's perfect then, because you have the best of both worlds.
Very much, yeah. I didn't mean to pose it as an either or thing. Like you said, really you want both ultimately. But now I'm going to push it and say something even more controversial. You mentioned CiteSeq. So CiteSeq obviously can help you interpret or profile proteins and isoforms that you can't easily do with single-cell RNA-seq, post-translational modifications, like you mentioned. But CiteSeq also has the advantage that you can profile proteins that are more abundant than the corresponding RNAs. So there's many well-known protein markers whose RNA expression is too low to be detected easily in single-cell RNA-seq. And that is one major advantage of CiteSeq. But as single-cell technology gets better and better, as single-cell RNA technology gets better and better, more and more sensitive, that gap shrinks. So the number of markers that are undetectable in RNA becomes smaller and smaller and smaller. So can we argue that the delta incremental value of CiteSeq becomes smaller and smaller as the RNA sensitivity improves?
So I agree with you, Shyam. So I think sensitivity is one thing, but the other big power of the sequencing modalities is the breadth, just the sheer breadth of data, right? And that's it's hard to see how you would get that at the protein level in quite the same way. And it's breadth and coverage, I would say. So sensitivity is one thing, breadth and coverage is the big strength that I see. Then the combination of multiple modalities, so that the epigenetics, the expression, and so on and so forth, is something that it's hard to see how you'd achieve that with protein, and then the other thing is the spatial resolution and the very high spatial resolution. In terms of breadth and multiple modalities, and that's where the sequencing technologies are a real strength, in my opinion, and just so powerful.
Absolutely, I think we're really at the cusp of this great change in immunology, and works like what you presented here are driving that. I wanted to ask you quickly about the second part of your talk, which is the limb development, and you just touched upon the mouse and human difference, which is very profound. And limb development is really one of the definitive aspects of primate evolution, one of the earliest aspects of primate evolution, apart from the fact that the eyes and the visual cortex became more important in primates than in non-primate mammals. And then, of course, throughout primate evolution, all the way coming to apes and coming to the human lineage, the limbs have evolved faster and faster, or diverged more and more, made us who we are. So I was wondering, do you have a third group to polarize the differences between mice and humans, like a primate group or an out group or something like that?
Okay. Absolutely. I mean, not in the limb, but yeah, I completely agree with you that from an evolutionary point of view, yeah, you want to have a much a broader coverage of the tree, and like you're saying, that may be different for different systems in terms of, you know, which part of the tree of life you're interested in covering, depending on the system that you're studying, as it were. Yeah, so I completely agree, and I think the cross-species studies, you know, will be so exciting in future. Yeah, as we get more dense coverage of data of different systems across different parts of the tree of life.
Quite so. And along the same lines, are you looking at the opposable thumb and things like that? Any plans to investigate the gene expression basis of, you know, that part of our limbs that becomes a thumb?
That's a great question. Yeah, we've discussed that actually. You know, that as humans we have that unique capability of this pincer motion, like with all the fingers, and you know, the orthopedic clinician, you know, and with them we've discussed that. No immediate plans, I would say. But I you know, I do think there's a lot of benefit from studies of non-human primates, and it would be nice if the you know, if it were easier to, I think, to gain access to those tissues, and for us to be able to study them more easily. I mean, there's a lot of challenges in that respect, I think, here in the UK, maybe less in Asia.
Yeah, even the human data set will be tremendously valuable for that. A couple of quick questions in the chat from Jay Shen. Great talk. Can you expand on the differentially expressed link RNA between human and mouse, and are they synthetically conserved between human and mouse?
Yeah, that's a great question. So some of them are indeed sort of antisense RNAs that are, I think, are largely conserved. And so it is, you know, it isn't obvious why they're species-specific expression, and it will be interesting to go into that in more detail. Yeah, no, thanks for that question.
Okay. And a question from Al Forrest also. Can you expand? Oh, wait. Okay, that's the same question. Never mind. I think copy-paste error. Oh no, I think two people copy-pasted the same question. All right. I think we're a bit over time right now. So thank you so much for staying with us, and for a terrific tour of the latest that's happening in the Human Cell Atlas. And thank you again for supporting Human Cell Atlas Asia.
Thank you for organizing this fantastic meeting, and have a great evening, everybody, and bye for now.